Hi, I'm Sarbjeet Niraula, a PhD candidate in Dr. Woo-Shuk Chang's lab from Biology Department. I'll be presenting a gene co-expression pattern of Bradyrhizobium japonicum under desiccation stress. It's a bacterium that forms symbiotic association with the soybean plant and fixes atmospheric nitrogen to ammonia and fulfill more than half of the nitrogen requirement of the plant. But this beneficial association is affected by the desiccation stress in the field and is a growing threat due to global warming. To mitigate this problem, we need to first understand how this bacteria behave under such condition at molecular level. So the goal of this study is to construct and analyze the gene co-expression network to identify novel gene or pathway uh, that are activated under desiccation stress. So the basic idea behind this study is that there are some genes that are transcribed in large numbers, or in other words, called highly expressed in certain condition, whereas some genes are less expressed. So we make use of these expression values to correlate many genes and form a network where the connections represent the strength of the correlation. Since the network of thousands of genes is very large and difficult to handle, we divide them into small groups of highly connected genes. Here we call them modules. So in this study, we use the expression values from the bacteria was grown under desiccation stress at different time points. Like all other biological networks, in our network also, there are few genes that have very high connections, and there are many genes that have less connections irrespective of the size of the network, which essentially means the scale-free topology. After verifying the scale-free topology, the modules were divided into 38, the network was divided into 38 modules of highly connected genes, and the modules with very similar expression patterns were merged and refined to 16 modules represented by different colors here. 5 out of 16 modules are positively correlated with the desiccation time, and the genes in each module share a common biological function as identified by the functional enrichment analysis. This also indicates that the modules are biologically meaningful. We further correlated the gene significance and module membership of each gene within the module of interest and found that the gene having high gene significance also tend to have high module membership. And these are the genes which play a central role in each module and are considered as the hub genes. We also found that the connectivity pattern of genes in each module are largely preserved in the large network formed by the gene expression of Bradyrhizobium japonicum from many other experiments. This suggests that the genes in these modules are actually part of a certain biological pathway. Finally, we plotted the network modules to identify the candidate genes, and here genes in purple color represents the candidate gene for further verification, which have high module membership, high gene significance, and have large number of connections. In conclusion, we showed how Bradyrhizobium japonicum respond to the desiccation stress with the genes as modular structure. And also we found some key genes responding to the desiccation stress that may help in the development of bioinoculant in the future. And currently we are validating these candidate genes using qRT-PCR and also by constructing the mutant and testing them on the desiccation stress. Thank you.